Oh, thank you. You're welcome.
Hello, everyone. I'm gonna go ahead and get started with the main program now. And again, thank you all for making it out, especially how harrowing the journey must have been for you to get out here. But as I was saying earlier, and this is great mushroom weather, and by the abundance that we have on the table over there, it seems like the start to a very promising season. So this is the November meeting of the LA Mycological Society, and from October through May, we meet here on the third Mondays of the month, and we are always glad to share with you a different featured speaker, in addition to some mushroom shenanigans beforehand. Some general announcements. First off, just be aware that we don't use microphones in here, and voices carry pretty well. So if even if you think you're whispering, just know that voices amplify pretty effectively, so a lot of people can hear you. So secondly, I just want to make an announcement that in addition to the regular programming that we do, like I said, from our season from October through May, February is our main event, when we host our annual mushroom fair at the LA Arboretum. Vanessa, would you like to say more about that?
Yeah, okay, so for the mushroom fair, I don't know if you guys have ever been before, but we serve food at the mushroom fair. If you guys are talented cooks or want to volunteer in the kitchen making the food that we serve at the fair, I'm going to pass this sign-up sheet around. We just want to recruit kitchen volunteers early because you will have to do a Serve Safe certification. So it's, I think it takes about an hour to do, but LAMS will reimburse for whatever cost the certification charges. So we want to give you time to get certified to work in a kitchen. And then as well for the fair, we have a mushroom art and photo contest. And we have some flyers over here with the rules. Pretty straightforward. If you have mushroom photography from the SoCal area, and it's a SoCal growing mushroom, you can submit your photos of that. If you have any art, you can submit one art piece per person. Everyone who brings their photographs and their art to the fair, they'll have a chance to have it displayed and voted on. And we have prizes listed on the flyers as well. And if you didn't get your prize from last year, come find us.
Yeah.
That's pretty much it.
And also, the mushroom fair is a completely volunteer-driven event. The way that attendance to the mushroom fair works is that you usually have to pay for admission to the LA Arboretum, but volunteers are able to come in for free, and you could volunteer for your allotted scheduled time and then enjoy the fair for the rest of the time. But yeah, it's a nice thing to be a part of in any case. And again, that's in February. Is there anything else from any other council member? Yes?
So if you're like a membership for the LA Mycological Society, is that helps to have a discount?
Well, having a membership to the LA Mycological Society it gives you access to our newsletter, which is a nice document that we have a very wonderful person working on, Meridian, who's not here today. And we also notify you when we have our forays, but you could get a discount when you're paying for merchandise at the fair, but the LA Arboretum handles admission on their own. So the LA Arboretum is a county property, and they're pretty strict with how they run things. So we operate by their rules, and they're independent in how they handle additions to that kind of thing. But anyway, I'm very glad to be able to introduce our featured presenter today, who is newly minted Dr. Arik Joukhajian, who is joining us from UC Riverside, where, as I alluded to, he recently completed his PhD in Sidney Glassman's lab, and he's here to share with us about fungal responses to wildfires, which is a big question that we've gotten from a lot of people, and I'm sure there's a lot that you'll take away, especially as you think about what's happened in your own backyards. Who of you have been affected? But I'll again, just one more. I'll say again that voices carry very strongly in this auditorium. So if you think you're whispering to your neighbor, the rest of us can hear. So let's give our respect, and I'll leave it to you.
Thank you, everyone. Happy to be here. Again, my name is Arik. I was in Sidney Glassman's lab at UC Riverside. But before I started my PhD, I was coming to a lot of LAMS meetings, so I'm always thrilled to be back. And if you were here last year, you might have heard me talk a little bit about my dissertation work, which was these 3 chapters about eastern Joshua trees and mycology in the Mojave Desert. However, this is basically, today this is a slide about all the things I'm not going to talk about, because my dissertation is a little too fresh for me to jump back into, so instead I'm going to talk about all the cool papers that I learned about along my PhD journey, and talk about the things I didn't get a chance to talk about during my dissertation. But just to give the background, my work during my PhD was on mycorrhizal fungi that associate with the eastern Joshua trees, as well as the impact of the dome fire, which burned a large eastern Joshua tree forest in the Mojave Desert. And my 3rd chapter was a comparison of fires in the chaparral and the desert, and how that impacted microbes using this network analysis or graph theory approach. And a little bit more about my fungal expertise. I got into mycology just looking for mushrooms and generally being satisfied getting things down to a genus level identification, unless it was something edible, like chanterelles that I found up in Mendocino County. However, when I have a mushroom in mind that I'm looking for, it's very easy to get tripped up by other lookalikes, and side by side, these 2 mushrooms don't look all that similar, but when you're zeroed in on finding something, finding this Hydropteropsis might throw you off into thinking that you found the chanterelle. So you always have to be diligent into when you're considering eating something you just found. And these aren't my bite marks. This mushroom came pre-bitten when I found it, but just to be mindful of those things. But my background is much more in the lab as far as mycology goes. So I do things like staining plant roots in order to identify mycorrhizal structures, and particularly this method is for arbuscular mycorrhizal fungi. And arbuscules are these little tree-like structures that fungi will make inside plant cells, and they're tree-shaped, and so that's why the name is arbuscule. But these are sites of nutrient exchange that the fungi creates in order to provide the tree with, or provide the plant with nutrients that it has access to that the plant might not necessarily reach. And beyond that, a lot of my work has been focused on sequencing microbes from the soil. So often what this looks like is getting a 1/4 gram of soil and then just doing a DNA extraction and amplifying the DNA to target an organism of interest. So if you're looking for fungi, you might use fungal-specific primers. And so this brings us to kind of the sequence revolution that changed how biology is done. So when we're interested in a particular group, we'll have to use a barcode region specific to that group. And so for fungi, this is the region, either ITS or 18S. And depending on the group, you might target different sections of DNA. But well, what a barcode is basically this region that's somewhat consistent and then has more variability as you go, so that you have something to look for that is consistent across fungi, but then also has variability so that you can differentiate different species of fungi using this DNA approach. And so what we do is use this region that is consistent across fungal groups and use these primers which will bind to those consistent regions, and then we can mass amplify that so we have these barcodes that are these regions of variability that allow us to identify and differentiate one fungus from another without ever having seen it. So this allows us to use these molecular biology approaches to identify fungi from the soil. And so this plot here is a comparison of basically all my samples when I do a sequencing run. I have dozens of samples, and I'll get different amounts of sequences per sample. And so here the average is about 100,000 sequences of DNA per sample that I'm getting. And what we do with that is match that to the Unite fungal database. And there's different databases for different groups, but this is the largest one and most broad one. So Unite actually has species hypotheses for 200,000 different species of fungi. And this is thanks to all the taxonomists that have cultured a fungus, identified it, described it, and then sequenced it. So we have this database that is well curated and has this large data set where we have tons of different species. So here, Dicaria, which is that combination of ascomycota and basidiomycota. This is basically all the mushrooms you'll find will be this chunk of blue that makes up most of the sequences in Unite. However, there's tons of organisms that are considered fungi that don't fall into this mushroom-forming group that are represented in this Unite database. And when we do these sequences, we're basically looking at the proportion of one fungus to another. And so this is looking at phylum level. So in these samples, which are soil samples and pine tree samples, we're seeing that this green color ascomycota makes up the dominant slice of these pies. But these circles are kind of hard to look at. Often what I do is turn all my sequence data into these bar plots where we can compare different treatments like burned and unburned at 17 days after a fire, and then you can look at the color of the different fungal genera and try to eyeball whether or not they're different in one group or the other. And the reason why this is mostly gray is because these are all the dominant taxa. So if I showed every fungus in each treatment, this would be hundreds of colors long, and you just can't look at that. So it's kind of for everybody's benefit to only show a couple different species, and you really want to focus on the ones that are dominant. So here we have this orange color alternaria, which was very common in my desert samples. But this is just an example. And so now I'll get into my familiarity with wildfire. So I'm from Pasadena, and went to Pasadena High School. Definitely saw a lot of friends and family affected by the Eaton fire. And in fact, I drove through Altadena getting here today, and it was very devastating for the community. But what we can see is that the fire was mostly in the hills above us, and it encroached into human development. But it's very common for the kind of standard ecology of California habitats to burn at kind of this rate, and these areas hadn't burned in some time. So, you know, after thinking about the impact on the community, I was also impacted by, you know, all the places that I like to explore in nature. This is my iNaturalist map of all the places where I've gone mushroom hunting and all the local trails. So it's definitely a loss to lose these places, but it's also part of the natural fire regime of the area. So it's kind of to be expected. Now, the last, or at least the 2010s were particularly rough for wildfires across California. So 2018 had some really bad fires, but 2020 kind of blew that out of the water with 3.3 million acres burned across California. And if we look at the monetary damage of the LA wildfires compared to this 2020 year, we see that they're actually neck and neck in terms of how much damage was done to human structure and development. And actually, if you look at the Wikipedia page, they've actually got this map of this chart of the 20 most destructive California wildfires since 1930. And the majority of them are from the last 10 years. And I actually looked at the source of this, and it showed that 9 out of 20 of these fires were caused by power lines, while only 2 out of 20 were lightning strikes, which often happens in California forests. And that's how the dome fire that I study in the Mojave Desert started. But you know, we're seeing a lot of consequences for our development. And in this map that I showed earlier, we've got another fire that's grayed out. This is the 2020 Bobcat fire. And while I was starting my PhD in 2020, you know, I was living in Pasadena and commuting to Riverside, and so I was witnessing this 2020 Bobcat fire starting, and I was watching it, you know, as it developed. And this is a view from my parents' house, and this was, you know, seeing it approach at night. And because I was working, I was already working with the Glassman lab, which studies fire and fungi. I was taking note of all the ash that was raining down on my parents' house, and so I took some petri dishes home from the lab. And these were dishes that were treated with antibacterial or antifungal compounds so that we could isolate some microbes from the smoke and ash that's raining down on us. So I held these petri dishes up in the air for 2 minutes and tried to collect some ash. And then we got all these cultures that have, you know, mixes of different bacteria or fungi depending on the plate. And then I tried to isolate these and purify them so that we can look at one species at a time and try to describe some of these fungi that are being spread by the fire.
Nice work.
So now a little bit of history about fire and fungi. I saw this very interesting paper from M. Cat Anderson and Frank K. Lake in the Journal of Ethnobiology. This is titled California Indian Ethnomycology and Associated Forest Management. And this is just a recognition of how indigenous communities use fire as a tool or a food source or medicine. And it's all listed out in this very interesting paper. And there's a quote from Katie Appling from the Southern Sierra Miwok or Pawite tribe, who says, "These crybaby mushrooms that are brown and shaped like teardrops come up after fires under bushes in the chaparral and manzanita." And so this is talking about these morels that have this teardrop shape and are commonly sought after by foragers. And in this paper, we've actually got this map of different tribes and those that are known to use mushrooms in different ways. So it mentions food, medicine, and other technologies. And this is a photo of Melba Beecher from the Mono tribe, which I've starred here in the middle. And she's holding a Lyophilum mushroom, which is a edible called the fried chicken mushroom. And we actually will see Lyophilum pop up in some of our my future slides. But she's got a quote in this paper where she says, They burn under the black oaks for better mushrooms in October, November, and December. The mushrooms that come up better are kayaki, which is this romaria. She mentions naka, which could be these Piziza sylvestris, and tupo, which is the Cacora or Amanita calypsoderma. And another person, Lydia Beecher, mentions kuyu come up better after a fire too, and that refers to these morels that we mentioned earlier. And then in the world of academic journals, we've also seen recognition of post-fire fungi as early as 1909. So Fred Seaver was the editor of Mycologia, and one of his first papers as editor was this paper called Studies in Pyrophilous Fungi, the Occurrence and Cultivation of Pyronema. So Pyronema is a common post-fire fungus, and in this paper he says, sterilization of soil by heat apparently brings about some change in the soil of vital importance in the cultivation of fungi, which normally grow on burnt soil. And so he's recognizing that there's this post-fire response by fungi, and there's supposed to be a picture of it, but this is what it looks like taken with a more modern camera. So you get these rivers of Pyronema fruiting and making their ascocarps after a wildfire. And so we see a lot of adaptations to wildfires talked about in the world of botany, and a very common one is the pine cone. So we mentioned the serotiny or pyrесcence of pine cones, and this is the fact that when you have the pine cone burn, some species, their pine cones will open up only when heated to a certain degree, and this basically allows the seedlings to sprout in a world that potentially has a cleaned up canopy. You might have more access to resources, you might have less competition in the post-fire world, and so these certain pine trees have adapted to California's fire regime. And then this was cool enough that I just had to throw it in there. There's this concept of firehawks, where in Australia these various different birds have been observed picking up sticks and spreading fire when wildfires occur, and this lets them pick up the rodents that are fleeing from the wildfire. So it's just an easy food source. When they see a wildfire, they know to spread it and get this easy snack. And it's a variety of hawks. I'm not sure what the spread of different birds is, but it's a very cool adaptation. And so in the world of botany, there's a lot of ecological theory, and one thing that's been adapted is the CSR triangle by Grimes. And so this is basically a matrix of where traits lie when you're adapting to something. So when there's a lot of competition, a trait might allow a species to outcompete another. And when there's a lot of stress, a trait will probably fall in this corner and give a microbe or any organism some mechanism of tolerating that stress. And then when you have a disturbance come through and change the landscape, you have traits that allow ruderal species to basically rapidly recolonize or bounce back very quickly. And so this has been adapted by a former lab mate of mine, Dr. Dylan Enright. And so in one of his papers, he shows the CSR triangle that's been adapted to pyrophilous bacteria and fungi, but I'm only going to talk about the fungi today. So in the stress-tolerant corner, you have these traits that allow you to tolerate stress. So you have these thermotolerant sclerotia, and you have spores that are resistant to heat. This is basically the pine cone that we saw earlier. And this is common in fungi like Pyronema and Neurospora and Rhizopogum. And then in traits that make you more competitive, we have post-fire resource acquisition. So this is being able to break down that post-fire environment and access some of the more complex things that are left over after a fire comes by. So fungi like Biophyllum and Pholiota and Penicillium have traits that allow them to outcompete other microbes and get to these post-fire compounds quicker. And then in the ruderal corner, there's the fast colonization or fast growth. And so this is things like a quick hyphal extension that allows the fungus to dominate in the post-fire landscape and rapidly expand into a niche. So many molds like Penicillium and Aspergillus will show very fast growth rates. And so I got sent a paper recently that was explicitly testing these concepts. So Neurospora, or specifically Neurospora crassa, is a famous model organism that is responsible for a lot of breakthroughs in biology in general, but it's well known as a pyrophilous fungus and has this fire response. So one study wanted to look at whether or not it actually prefers these burned substrates or if it just gets to colonize these areas quickly. And so they actually tested this with aspen wood, spruce wood, and corn stalks. And so these are these three substrates that they're using, and then they had an unheated one, and then one that was heated up to 225 degrees, and these blackened ones were heated up to 350 degrees. And they tested these three fungi on which substrate they preferred and how much degradation they actually did. And when we look at it, we see that Neurospora basically didn't degrade any wood at all, ever, and in fact it had negative mass loss, which they say is just the fungus growing on the wood and not actually breaking anything down. But what it could do is break down this corn stalk, and it didn't necessarily break it down any better once it was heated up. So they set out to test whether or not Neurospora was preferring burned materials, but what they actually see is that Neurospora might happen to be heat activated, but really it doesn't have anything special to break down these burned materials. It just grows really quickly. And we actually see proof of this in my former lab mate Dylan's work, where he showed that Neurospora was really good at growing fast, both on regular MYA media and this burned media that he made with ash taken from various fires that we've collected from. And he also shows that Neurospora discreta had really bad enzyme production rates, so it's not doing anything special in terms of accessing plant carbon or nitrogen. It just grows really fast. And so these are just some different species of Penicillium, which is a mold that we collect very often from these burned environments. And I point all these different species out because in another study looking at wildfires across Canada and northern United States, there's this paper by Thea Whitman that you could read on bioRxiv. It's called Resilience Not Yet Apparent in Soil Fungal Communities of the Boreal Forest from 1 to 5 years after a wildfire across the severity gradient. Okay, so severity gradient is just how bad the fire killed plants. And so you have these highly severe fires that are marked in red in terms of how much Penicillium species you got, with orange being not so severe fires, and yellow being unburned forests. And basically in this paper, they're showing that these two Penicillium species did not care whether the forest was on fire or not. However, when you look at other Penicillium species, you do get some species that really prefer these wildfire environments. And so we have this strong preference for the burned state in certain species and not others. And so there's kind of this variation across species within the same genus. And so you kind of have to be mindful of what species you're looking at when approaching these kind of post-fire environments. So one other concept is competition, which I mentioned. And so instead of developing a gene that makes you outcompete your neighbors, you can also just access bacterial genes as a fungus. And so this is called horizontal gene transfer. This is when fungi will just take up genes from the environment and use them under certain circumstances. So this is another study from the Glassman lab, from a scientist named Dr. Arik. And so we have this gene, catechol 1,2-dioxygenase, and this cat-A gene helps this Penicillium atrosanguineum break down catechol, which is this common post-fire compound. And in this study, they're basically showing that you have these different genes for this cat-A enzyme, and when you have a normal gene that you've developed, you often get mutations that are synonymous. So these are mutations that don't change the enzyme at all, and those are listed in blue here on these blue bars. And occasionally you might get one non-synonymous mutation that actually changes the outcome of the gene and allows you to outcompete other microbes. However, these genes listed in red have way more of these mutations that actually make this enzyme work better in the post-fire environment. And the thought is that these genes mutated under completely different circumstances from the rest of the genome. So these genes that have these really high orange bars, these are things that this fungus, Penicillium atrosanguineum, probably just took from a bacteria and then used it to outcompete other fungi in the post-fire environment. And so I've got one other paper on this topic that came out earlier this year. This paper goes over the idea that, you know, there's tons of papers suggesting that there's bacterial genes being transferred to fungi, and this paper looks at 400 of these cases and says that, you know, if we take this conservative look at it, probably only 1% of these are actually real, where the gene started off in bacterium and ended up in fungi. And I wanted to focus on one of these cases where this gene transfer might be relevant to these post-fire fungi. So we have this gene transfer that happened in the ancestor of these three fungi, Lufia, Exophiala, and Cladophyllophora. And these are fungi that I often see in my desert data sets. So these are melanized ascomycetes, and they're very common in these semi-arid areas. But they're also very well researched because they can be human pathogens. And so people who are immunocompromised can see, you know, black patches of these fungi growing on them because they have these melanized exteriors. But the gene transfer event was this gene for alcohol dehydrogenase, gross like domain, and basically it helps break down toluene. And toluene is a hard to break down compound that not every microbe is going to be targeting. But by acquiring this gene from bacteria, these fungi can use this post-fire compound as a food source. So this really gives them the ability to outcompete their neighbors by having this gene acquisition within the happen. So now I'll talk about mycorrhizal fungi. And as you can imagine, this group of fungi are very sensitive to wildfires because mycorrhizal fungi, and particularly ectomycorrhizal fungi in this case, associate with a lot of plants and are very common in these large forests that often burn. So when you have a fire come through, it can burn the host, and if it gets hot enough for long enough, it can also burn the fungi below ground that are associating with the plant roots and transferring nutrients from the soil to the plant. So looking at one study by another Glassman Lab member, Dr. Fabiola Pulido Barriga, her dissertation work focused on the Holy Fire, which is near Temecula in the Cleveland National Forest. This is a very dense shrubland area, and it's also got pines and oaks, so it's got tons of things for ectomycorrhizal fungi to associate with. And this wildfire burned very intensely when it came through. And what she did was go back week after week and collect soil from this wildfire and look at the different sequence abundances of fungi that are present in the soil. And so we see a couple different fungi have really different trends. And so Pyronema, a common post-fire ascomycete, goes from nearly zero to being the majority of the sequences in the post-fire soil. Meanwhile, we have ectomycorrhizal fungus ionosobi that, makes up around 20% of the sequences when you sample some soil, and this steadily drops over the course of 1 year. And so they're really disappearing. And this data set now goes out to, I think, 7 years. So there's a lot of info that we have on these different microbes over time. But just to look at it from a different angle, here's another study from another former Glasson Lab member, Dr. Marcos Caiafa. And so in this paper, the group was sampling these beetle-killed forests across Colorado, where you have most of the trees already dead, but you do have, something like 20% of the trees surviving in the forest. And what they did was, after many of these forests burned, they went and sequenced soil from these burned and unburned forest areas, and they looked at fungal abundance across Colorado, basically. And so in this plot, what I'm showing is a bunch of different fungi on the x-axis, and if they're above 0, that means they really like the fire and they increased in sequence abundance post-fire. And if they're below this 0 line that's dashed, that means this particular fungal isolate didn't like the fire and was more abundant in the unburned areas. So when you compare the burned and unburned areas at this forest sampling event that happened 3 years after the fire came by, we see that various fungi were much more abundant in these burned plots. So this first one is Paraphoma. We have Geopixis, these ascomycetes cups. We've got Foliota, that's common Basidiomycete, mushroom you'll see popping up after wildfires. But if we look at the back end of this graph, we see basically all these different ectomycorrhizal fungi, like ionosobi, which I mentioned earlier, Tomentella, which doesn't form mushrooms, it forms this crust, but still makes these ectomycorrhizal relationships with the trees. And we also have Cortinarius down at the very bottom. So, even 3 years after fire in these forests that are nearly dead, the fire is still heavily devastating these mycorrhizal fungi. And so this can be really rough for the forest to come back, but there's other fungi that I'll talk about that might make up for that. So we also see a lot of mycorrhizal fungi show stress tolerance, and this is work by my advisor, Sydney Glassman. So Sydney was studying ectomycorrhizal fungi and their spore banks in Yosemite in 2011. And the way you do this is take some soil from the forest, bring it back to the lab, and then plant a sterilized pine seedling into that soil, and then you see which ectomycorrhizal fungi are infecting your seedling. And so this allows you to basically do a bioassay that tells you which ectomycorrhizal fungi are active in a certain forest. Unfortunately, the places where she collected soil from burned down in the 2013 rim fire. And so, maybe fortunately, they now have this very rare data set where you have pre-fire data and post-fire data, because all the other fires I talked about earlier were a comparison of burned and unburned forests, but in this study they went right back to where they collected that soil. They took it after it burned and then had kind of this one-to-one comparison you can make of pre and post-fire. And they see this one mushroom, Rhizopogon olivaceo-tinctus, actually be one of the only ones that only showed increase when they did these sterile pine bioassays. And so Rhizopogon olivaceo-tinctus is this false truffle. So this is a truffle from the Basidiomycota. It's not technically the delicious truffles people are seeking out, but if it looks like a truffle, I'm calling it a truffle. And so Sydney's advisor, Tom Bruns, who called himself my science grandpa, because advisor, he's been studying Rhizopogon for decades. And so when he saw this result, he became interested in asking this question. Is Rhizopogon olivaceo just thermal tolerant, because it has this melanized structure that might tolerate the fire better than others? And is it just dominating the sterilized post-fire soil? Or is it actually responding to the heat and, is it heat activated? And so after the previous result, he set up this experiment where you take the spores of this Rhizopogon and you put them in these trays that you either heat or don't heat, and see how much infection you get. So when you have pine seedlings planted into soil with Rhizopogon spores that hasn't been heated up at all, you get about 50% of seedlings getting infected by this truffle. But then when you heat up the soil to around 50 to 60 degrees, which you can expect a couple centimeters below ground, where these truffles are existing, this actually flips to nearly 100% infection of your pine trees after, just lightly heating the soil for an hour. And so it's clear that these truffles are not only heat activated, but also thermal tolerant. So now looking at morels, these show a more ruderal response. So these are the things that grow really quickly after a fire occurs. And this has been recognized in the indigenous knowledge that I showed, and also modern day forager knowledge. You can watch videos of people like Northern Wild Harvest in British Columbia going to forests after fire and collecting tons of morels. And I just have a plug for my own art piece of after the 2018 Woolsey fire in Los Angeles. I didn't find any morels, but I had, an idea of what kind of suspect we might be looking for. So this is just a reminder to make a submission to the art contest for the fair. But I wanted to try to think of, how are these morels actually growing so quickly in the post-fire environment? And I looked up, they have something called radishes sclerosum. And so I don't know if any of you have ever found this when picking morels, but you'll often see this root structure in certain species. These are just very thick hyphae that basically serve as energy reserves. And so once a fire comes through, they actually already have a lot of stored energy to rapidly throw up these fruiting bodies. And the point of the morel mushroom is to spread the spores. And cup fungi often appear in these post-fire environments, which, if you can imagine, after a fire, you've got all the brush cleared out, you've got potential clearance in the area, and so, with one good breeze, you might release a ton of morel spores that are now being spread across the environment. And these are just some color photos of another morel, Morchella norvegiensis, and just to show that, it's not necessarily just one species of morel showing this kind of trend. And one other idea for explaining how certain fungi are very ruderal and resilient and able to bounce back really quickly post-fire is this body snatchers hypothesis. This is a paper called Where Are They Hiding? Testing the Body Snatchers Hypothesis in Pyrophilus Fungi by Radabaugh et al. 2020. And in this paper, they propose this idea that many fungi are just living harmlessly as endophytes inside plant cells. And this is a common form of existence for fungi where they just harmlessly live inside a plant, and these are fungi that aren't necessarily causing an infection, but might just be waiting for certain conditions. And for some of these fungi, you get existence living inside a moss, waiting for a forest fire to occur, and then you start growing your mycelium, and after a couple months or a couple years, you might fruit your sporocarps, your fruiting bodies, and your mushrooms, and you might continue this cycle. And in this paper, they talk about, or they identify several of these pyrophilous fungi, like the Foliota mushrooms, the Geophysis cup fungi, as well as morels and pyronema. They found these using both sequence and culture-based methods. They found these growing in mosses and lichens that they were sampling. And so there might be some truth to this idea where these fungi are just living as, they're like waiting and just biding their time for a wildfire to occur so that they can then fruit and put up their fruiting bodies. And so we actually saw some good evidence of this in the dome fire in the Mojave Desert. So when my advisor, Sydney Glassman, first went out to the desert, she noted tons of neurospora growing on burned Joshua trees. And this turned out to be Neurospora discreta, which we had confirmed by culturing and sending to a Neurospora scientist, Pierre Gladeau in France. And even 8 months later, after a fire, this Neurospora was still, apparent, growing basically under the bark of these Joshua trees. And actually, these sequences that I showed earlier were sequences from my soil data set, where I was basically collecting soil from under these Joshua trees. And these belong to different Neurospora isolates or unique sequence variants. And I found Neurospora terricola, Neurospora canonica, Neurospora dictyophora, but we sent these sequences to Pierre in France, and he said none of them match the tree, colonizing Neurospora that we were seeing. So this potentially gives some credence to this body snatcher idea, where these fungi in the soil don't match the fungi in the tree, and so potentially the way these colonize these Joshua trees so quickly after the fire is by simply living inside either the bark or the leaves of these Joshua trees. And as soon as a wildfire occurs, all their competition has been burned off, and they can then colonize this area and create these fruiting bodies. And just one story related to this, I was actually going to the Mycological Society of America meeting in Phoenix, Arizona, in Arizona in 2023, and basically the night before my lab group drove out across the Mojave Desert, we got word that a wildfire even bigger than the fire I was studying in the Mojave Desert had started. And so as I was driving through the Mojave Desert, I was seeing all this billowing smoke, and this 90,000 acre wildfire was occurring in the Mojave Desert. And in the time between going to the MSA meeting and doing a talk about my 2020 dome fire and this wildfire occurring, there was actually also Hurricane Hillary, which came through in this area and dumped a lot of water. As you can see, a ton of greenery. And let me hit play. So I eventually came back to this site and tried to see what had happened in September later that year. And I was making note of the Joshua trees, which are the species Yucca jaegeriana, and they also still had this Neurospora growth underneath their bark just a few months after the fire. But there's also other other yuccas, like the Mojave yucca here, growing just right next to it, and it has zero Neurospora colonization. And I was looking for, this Neurospora all over in the different Joshua tree relatives, like the banana yucca or the Mojave yucca, and I was exclusively seeing this Neurospora colonization happen on these Joshua trees. So I think there's still a lot to uncover about how fungi exist in the natural world, and this famous model organism Neurospora still has a lot of unknowns, and I think it'd be very interesting to find out that Where it's actually hiding in the natural world is in Joshua trees, at least for this one specific species. And so, just to summarize, the different mechanisms I've shown of responding to these fires are these melanized sclerotia and heat activation spores. These methods of unique gene acquisition that allow microbes to outcompete each other. These energy storage strategies that we saw in Morchella, as well as the body snatching strategy in Neurospora species. And so there's a variety of different traits that allow these fungi to respond to fire, but there's still a ton to uncover, and so that's what initially got me into mycology, and hopefully piqued your interest to look into some of these different methods yourselves. And so with that, I'll thank you for your time. And
we'll take questions now, and while we're doing the questions and answers, can we just continue to talk in zero and it's just take this thing along. So anyone has a question for Arik?
sorry, I'll start up front.
I'm curious about the species that you did put body snatching with. Was that then a new species that had been identified?
It had been described. So the species we found on the Joshua trees was Neurospora discreta, and it had been described in other places, and I think other people had seen it growing on Joshua trees before. What we did with that particular isolate was take different Neurosporas across different trees, across the large acreage of the fire, and we sent it to Pierre to do population genetic studies. I'm not sure what the outcome of that was, but potentially these are some that haven't been sampled because this area hadn't been burned in a while, but still to be determined. Yeah.
The bacterial gene transfer, you said that some of them were legitimate and some of them were not. How do you differentiate?
That's a great question. Identifying whether or not the different cases of gene transfer were real or not kind of goes above my understanding of genomics themselves. So they're looking at a lot of different signals that show where something might have originated from. There's certain signals you can look for in genes, like the content of G's and C's versus A's and T's. Those proportions can actually vary based on where an organism is adapting to, and I think that can be temperature related. And there's a lot of these little signals that kind of tell you this gene probably was mutating under completely different conditions. And I think what they've seen is that when you zero in on some of these trees, you see the same gene that has been gained by multiple different species and has potentially been lost in all these neighbors because it might have some, extra cost in keeping this gene. But I don't know the exact specifics that the paper was looking for in terms of calling one legitimate or not. Yeah.
Are any of them good to eat?
I'm gonna say no, but you never know. Any other questions? Yeah.
Yeah, is any of this research leading into current research on fire resilience?
Fire resilience in terms of the whole ecosystem, or?
Yeah, I think maybe for human or conservation efforts.
Yeah, I think a lot of folks have looked into this angle of can we bring in fungi that are maybe more fire adapted, and can we colonize our yards with it or something? There's some concern of spreading invasive fungi across the United States, which we're seeing with certain edibles right now. I don't know how you would. There's potentially, room for some beneficial fungi to be used agriculturally, but I think spreading things across the wild landscape will probably do more harm than good, just because a lot of these things have been adapting much longer than we've been paying attention, and so the fungi in these areas have experienced these fire regimes for a very long time, and some people are investigating how these different things are resilient in different ways, and how one microbe might tolerate drought better than the other. And there's a lot of interesting research about that at UC Riverside. But I don't know if anyone is encouraging jumping the gun and spreading some of these things quite yet, before we really understand the full picture, which we're a long ways away from. So it's a good thought. Yeah.
What about maybe the heat of the fire destroying some of the competition, essentially giving a pasteurizing effect on the substrate? Is that considered as a possible reason for things flourishing after a fire?
Yeah, so in this paper about Neurospora, what they're suggesting is that, some of these fungi that don't necessarily have any obvious traits that make them better at dealing with burn things specifically, these are taking advantage of that sterilization of these different compounds. So Neurospora probably has some heat tolerance compared to some of the other fungi that normally exist where it colonizes. And yeah, the idea is that because maybe the bark of the Joshua tree has been sterilized, now Neurospora with its really quick growth can jump in and quickly colonize the tree and take advantage of that empty niche. So yeah, that's what this paper is trying to suggest, for sure. Yeah.
You mentioned it showed the picture of the morels with the very thick mycelium.
Mm-hmm.
Do you know if they refruit on the same place, or if they grow a new one every time?
I'm not sure. I think that might lend to that, forager tradition of always cutting the morel at the base. I'm not sure. As far as I've seen, people have done studies on picking morels versus cutting morels, and I think they've got the same flush the next year. But that would make sense that if you leave it behind, potentially you can get another fruit out of it. So I don't know that the papers I looked at considered these kind of hyphal reserves that these morels are fruiting from. So it adds another layer of something to consider in these studies. Yeah.
For the Neurospora that grow inside the Joshua trees, are they if they're just laying dormant, are they detectable anywhere before they fruit, or you can see them?
Yeah, so the Joshua tree, Neurospora, could be detected probably. You would just have to know what plant tissue to look at, because we don't necessarily know if they have a preference for leaves or the bark or where they're coming from. But there's a lot of studies on endophytic fungi and how they just exist nearly everywhere. And there's also, I think, the epiphytic fungi that just live on the surface. So these endophytes are literally in between the square plant cell walls and just growing harmlessly in these plants. But there's also things on the surface of leaves and surface of structures. And there's a ton of research on this. You can look up Betsy Arnold, who has done a ton of endophyte fungi studies. And yeah. Any other? Yeah,
So when you were doing those air plates during the fire and falling ash,
not sure that we picked up any human pathogens doing these culturing studies. I'm sure, I think there's kind of like a spore density effect. If you're like breathing in a ton of neurospora, it just colonizes plates so quickly that it's just like growing so fast. But I don't remember getting anything I was particularly concerned with from these plates, and I would have to look at my notes to identify the actual antibacterials and antifungals we were using in these plates, but I can get back to you on that if you want.
The horizontal gene transfer, when they like steal that gene from the bacteria, is that when they reproduce, is that like passed on to their future generations, or does like each individual have to keep doing it?
Yeah, so I think there could be cases of horizontal gene transfer where it just stays for one generation, depending on how costly it is to keep that gene, or potentially the method where they acquired it. But what this study is suggesting is that there's these 8 cases where a fungus took a bacterial gene and incorporated it into its own genome, and that can be detected across its future species, because these events happen earlier in the phylogeny of these different species, and so that's how they're kind of identifying how these different transfers occurred, by seeing the same gene in multiple different species. Things like Penicillium and Aspergillus sharing the same exact gene that maybe looks different from the rest of all their genomes. So these 3 gene transfer events likely happened in some ancestor of Aspergillus and Penicillium.
Do you know if there's any efforts of utilizing fire-adapted fungi for like bioremediation efforts for contaminated soils?
I'm not sure if specifically fire-adapted fungi are being used in bioremediation. I know that, generally, people are having success with just the Pleurotus austriatus, the oyster mushroom. And in soil remediation, I think arbuscular mycorrhizal fungi are being used with other plants and microbes to have this kind of compound effect and benefit. I think for the large part, there's a lot of very useful plants in this regard. That if you've heard Danielle Stevenson talk at any previous LAMS meetings, she's done a lot of work on that sort of bioremediation using a mix of different things.
She's going to be speaking next month in December.
Yeah, so tune in for that one, and I'm sure she'll have a lot to say. But I'm not sure if there's any fire-specific fungi in that group. Yeah.
For mycorrhizal fungi in the soil, is it possible to track if it's survived fires before, similar to how trees you can see in rings?
That's a good question. I don't know if you can actually see signs of fire survival. Maybe if, it's similar to these morels, maybe if you have signs of these radichy sclerotium existing in the post-fire world, maybe you can see that it's been around for another fire, but nothing I've encountered. Yeah, I'm not sure. It's a good question.
So next week we are going to around Pasadena and the Eaton fires. I'm curious about you hike in the forest after these rains, what you can find.
In the post-fire environment, I've never seen morels in the Eaton Canyon area, but maybe this year this is when they come up. There's a lot of mushrooms like the ascomycetes that form a lot of these cup structures, the geopixis and blanking on the other cups. Pyronema, you can see. I actually haven't gone out to any of the burned areas in LA So I'm not sure what the, LA specific things, if they differ from, what we've seen in the Cleveland National Forest, but we've definitely seen these Foliota highlandensis come up after fire. I'm not sure if there's any, particular edibles or anything to look for, but there's a whole lot of these different pyrophilous fungi that you can generally expect. I'm not sure about Pasadena specifically though.
Thank you.
Does pyronema have a fruiting body, or is it just kind of like that mass there?
I think this is its fruiting body. I don't know if it particularly, I don't know what its lifestyle is before you see it like this.
It's kind of like a crust, like a cup.
Oh, the fruiting body is a cup, or?
Well, I think those are sexual structures, so I think those are still asci. It's just
Yeah.
like a very long cup.
Mhm. Yeah, there's a whole slew of cup-like ascomycetes that will pop up after fire. There's tricharinas and pseudo-tricharinas, and I think there's something similar to Geopixus that I'm blanking on. But yeah, I think growing in the post-fire area is useful for these cup fungi because of what I'm assuming is the access to the breeze, but that's just a guess.
Pyro doesn't mean fire?
Yeah.
Nothing? Yeah.
So it's kind of long since recognized as a fire-loving fungus, so to be named pyronema. Any other questions? Yeah.
Have you, I don't know if you mentioned, but have you guys tried using like liquid smoke to like simulate fires in your lab? Maybe with like native plant seeds, they'll use it to like break down seed code and stuff.
Yeah, I know we've tried to use liquid smoke to get manzanita to sprout for some of our studies. I don't know if we've used it in our mixtures. We have stuck with just using this very complicated method of breaking down charcoal and filtering it in order to get it into our pion media, as Dylan calls it. And so I'm not sure if liquid smoke ever goes into that, but it could help.
Do you mean liquid smoke like what you put on food?
Yeah,
Oh, wow.
yeah, it's been used for trying to get manzanita to sprout because they like the post-fire environment, and so it could maybe work in some of these. Yeah.
This is gonna be super basic question, because I don't really know anything about fungi, but, I've been very concerned about air quality, obviously, since the fires and all the volatile organic compounds which are not being tracked by AQFD, et cetera. But I'm curious what is known about how those sorts of, how air impacts fungi, in this particular setting, because this was a huge urban fire, so much toxic air.
Yeah, I know there's been work done on the differences of like urban fires versus wildfires and how that can affect microbes. I don't know the specifics, and particularly for this fire, I've been kind of checked out of the aftermath of the Eaton and Palisades fires, just trying to get my PhD wrapped up, but I'm sure there's going to be a lot of interest, in the coming years of people who have studied it recently. The work I've seen done is more focused on the waterways, so I'm not sure about what the impact of the air following these fires
Well,
is.
those waterways is an interesting question, because the heavy metals, the asbestos, like all that stuff, right?
Yeah.
I mean, how does fungi respond to that when it's in the water?
Yeah, that's being tracked by someone at UC Irvine. I believe his name is Chris Oliveras. He's a professor there working on this question, and I think it's a little too soon to know the results, but they've got some preliminary data, which I'm blanking on the results, but yeah, people are working on these questions. I Briefly saw the outcome at the conference, but I'm not sure where exactly things lie in terms of the impact on fungi specifically.
Well, about the air quality, I brought up this question about recurring pathogens. I just heard a thing on the radio that was talking about a recent paper that was relating some trend that was observed in some Bay Area hospital during the 2020 fires. But anyway, I mean, there's just so much more density of microbes when you're breathing in all of that ashy air. And the specific thing is that there's an increase in skin infections for people that are like recovering from a burn or even like a chemical burn. I have a colleague of mine who likes to clean glassware with acids and doesn't use gloves and he just accepts that he'll get burned sometimes. And then I told him this and then he said, oh, I've noticed that myself. When I burn myself, I get a fungal infection. So yeah, if there's a wildfire and you're recovering from a burn, you might get a skin infection at a higher likelihood than if there wasn't a wildfire.
Yeah, the thing I was concerned about was valley fever, coccidioides, fungal infection, and it's much more common out east, and I think even more common out in the desert. I briefly worked as a organ donor screening tech, and we would often get positive coccidioides results coming in from Riverside County. And so it was something to be mindful of. But I mean, I didn't take any particular precautions to deal with that. I just tried to not be immunocompromised and do that at work, I guess. Yeah. Any other questions?
I was just curious, did you get the Manzanita sprout with the liquid smoke?
I don't think we did in the lab, and I also personally tried it at some point and never had success, but I think you really have to treat the seeds before adding just liquid smoke. There's like different combinations of boiling water and adding liquid smoke and maintaining this over the course of like a year. I don't know how often you have to do like liquid smoke additions, but I'm not a particularly good botanist, so I didn't have any luck with it, although I think I still have some seeds lying around in pots that just never sprouted. Any other questions? Yeah.
All right, well, let's thank Arik for you're welcome to help yourself to some snacks and still look at some mushrooms on the table. We're out and cleared by 10 o'clock, so about another hour that we'll before we start packing everything up. But if you want any mushrooms from